Hi everyone, I'm Jérôme Arnoux. I'm a PhD student in the LabGem team, and I'm developing PANORAMA, a comparative pangenomic tools to explore interspecies diversity of microbial genomes. In the last decade, with the evolution of sequencing technology, there is an explosion of prokaryotic genomes available in databases. And now, prokaryotic genomic studies rely on the comparison of thousands of genomes for the same species. But they usually refer to one genome, and whereas there is a high diversity of gene content from horizontal gene transfer or polymorphisms, that's why since a decade now, there is a paradigm shift from reference genome to reference pangenome. But what's a pangenome? A pangenome is a union of sequence or genes from a set of interest genome. In our case, and in most of the recent tools, they divide the pangenome in three parts, with the persistent genome corresponding to genes that are common to all genomes or almost all genomes, the structural genomes corresponding to genes that are with an intermediate frequency between genomes, and the cloud genome, the genes that are specific to one genome or rarely shared between genomes. And with this new data model, we have a better representation of the genomic diversity, but we need development of new algorithms and tools for pangenomics analysis. And this pangenomics analysis can help to have better understanding of the adaptive potential of bacteria and of the evolutionary dynamics of prokaryotic genomes, because this evolutionary dynamics is linked to gain, loss, or gene modification. And in the gene gain, there is the horizontal gene transfer that correspond to exchange of genomic material between prokaryote species from different lineage. And this transfer results in a highly variable region that we call the region of genomic plasticity. And these regions are not spread all around the genome. They are grouped together in a spot of insertion. And thanks to the pangenome data model, we can detect and analyze this variable region um, with more efficiency. That's why in the LabGem team, they developed the Pangolin software to reconstruct and partition a pangenome graph. To do it, we have multiple genomes, and we will cluster the genes in gene families, and we will link them in our pangenome graph with the neighborhood relation. And after, with statistical method as the name algorithm, we can partition the gene family in persistent, shell, or cloud. And Pangolin now can use this partition pangenome graph to study the variable region with the PANRGP method to predict the region of genomic plasticity, and to detect the spot of insertion. And it can also study the colocalized and co-recurrent gene families in the pangenome with the panmodule method. So far, so good. We've got a tool to construct a pangenome graph, to analyze one pangenome graph. But now, what about comparing pangenome graphs? It's the aim of PANORAMA, the tool that I present today, to provide a new methodological development to compare prokaryotic pangenomes. PANORAMA is a Python software that can run pangenome analysis in multiple pangenomes from Pangolin in the same time. Today, I will present to you the genomic context search features and the biological system prediction feature, and I will give you an overview of the outputs available in PANORAMA. And another important feature that there is in PANORAMA is the comparison of pangenome based on all this analysis. So let's go to the genomic comparison, genomic context search. A genomic context is a conserved genomic organization which may be involved in the same cellular process. You have here as example, we have two target proteins that we will align to our pangenome gene families with the information of which gene families are corresponding to our target protein of interest. We can, after, extract the gene in a certain space of our different genome in our pangenome. We can also add a transitivity closure link between the gene in the different genome relative to our gene of interest. And thanks to this new information, we can reconstruct a subset of the pangenome graph with the information of gene family, neighborhood relation, and transitivity closure. But this genomic context is not corresponding to a genomic context conserved in the pangenome. We need to filter this graph. For that, we use a Jacquard index, and we keep only the strongest edges, and we obtain finally our genomic context conserved. But this analysis concerns only one pangenome at a time, and now with PANORAMA we can did it in multiple pangenome in the same time. So for one genomic context, we can predict the genomic context in multiple pangenomes. And with this, with a second information that is the similarity between family, thanks to a gene families clustering to obtain super genes families, we can obtain information of genomic contexts that are similar. We have a system of score threshold to say which system are similar to another. And at the end, we can say that we have a conserved genomic context between species. We applied this method on the hapiose degradation pathway. The hapiose is a sugar that is a component of plant cell walls and also a secondary metabolite of vascularized plants. And we searched the context of degradation in more than 1,000 pangenomes. We found it in 100 species. Most of the time is conserved and is not in whole the genus, is in specific genus as Echalaecia, for example, and that was a pre-work to identify new alternative pathway for the hapiose degradation in different genus. So let's move on to the system prediction in a pangenome graph. A system is a group of functional proteins that works together to carry out cellular processes. To predict a system, we need to have a model, which is an exhaustive and specific representation of the system. The model describes the gene presence, absence, and the synteny rules of the system. Currently, we integrate 461 models in PANORAMA with 435 defense systems against viruses and 26 secretion systems. We choose the JSON grammar to write our system because it's the more native grammar compatible with Python, and it also gives the opportunity to all users to write their own system for PANORAMA. And a system looks like this. You have parameters that are at the model's level. The model is divided in different functional units, themselves divided in families. Functional unit and families can be mandatory, accessory, forbidden, or neutral. And there are with several parameters also. And one of the important parameter is the maximum separation parameter, because we know that families and functional units must work together, so they must be in a certain space in genomes. So we have our models, we have a pangenome graph. Now we want to predict the system. So the first step will be the annotation of the pangenome graph with a HMM database to obtain an annotated pangenome. After, we can use the function of genomic context search to search our model corresponding to our system in our pangenome. We obtain different genomic context search that corresponding to the parameters of maximum separation. And the last step is to check if the parameters, all other parameters are validated, as for example, the minimum mandatory family or the maximum family forbidden. And this, with all this information, we can say which system is inside our pangenome. So we would to validate again our method. For that, we choose the publication of Flores Ramos and al. From 2021. In this publication, they study the Doloz-Ingolden pigram spaces to analyze different system, and they link the different system to spot of insertion. So we use 32 genomes based on the 28 genomes of the publication. In our study, we detect with pangolin 91 spots. With Panorama, we predict 209 systems for 28 different types of systems. And we could link our systems to 8 different spots. And in comparison to the publication, we retrieve all the different systems that is predicted manually in the precedent publication. And with Panorama, you can also have some automatic figures generated, as for example, this upset plot, which give you the information of which spot is linked to a system in which genome for which type of system, and the number of system per genome and the number of system per type. Now we would to do a benchmark of our method against two tools that did the same job but at the genome level, Padlock and Defense Finder. So when we compare our result to Padlock and Defense Finder, we retrieve the same systems than them, but we also have some additional systems predicted. But these additional prediction are linked to the fact that in Panorama, a gene family can be associated to several different systems, and that with the pangenome data model, the gene families integrate the pseudogene information. And we did the prediction of the system in 7 to 15 times faster than Padlock or Defense Finder. So now I present a work on only one species, only one pangenome. Now we are currently comparing systems among pangenomes. So for that, we choose the Acinetobacter genus. It's an interesting genus in our case because there is many spots and many systems in the different species, and we can easily link systems to spots. And our first result shows that with Panorama, you can easily find which system are commonly present at the genus level, and you can also say which system are specific to a species in your genus. And also, this figures is automatically generated. So in conclusion, today I present to you Panorama, a flexible and open source solution that enable fast and easy to use comparative analysis of pangenome. There is two important features, that is the genomic context search with the genomic context comparison, sorry. There is also the system prediction method with more than 400 systems integrating. We are ongoing to integrate the KEGG modules and the polysaccharide utilization loci to study metabolic pathway. And as I said before, all users can define their own systems with their own HMM to use Panorama to search a specific system in their pangenomes. And Panorama is an open source software, but for the time is not public. I hope to give you an alpha version soon, and we are waiting for that because we want to have a Conda environment linked to it to have all the documentation and everything to help everyone to use Panorama. I want to thank all the members of my lab and particularly the person who works on Pangenome project with me. And I have also a poster today if you want to come and to discuss more about these tools. Thank you for your attention.